lab manuals?
Make sure you have your lab manuals and bring them on Tuesday, and make sure before you go to lab on Tuesday you print out the stuff you need for Tuesday. Also, with respect to Moodle, I put the wrong files on there last time that had a whole bunch of extra stuff. I just cut out the stuff that you don't need, and some of this stuff, we're not going to be able to talk about every single chapter, everything in every chapter in this book. I could use this for like 3 semesters worth of stuff for classes. And so most of the time I'm going to try to cut out the stuff from the slides, because if you're inclined to print them out, that'd be a big waste of paper, because we're not going to get to all the stuff. Okay, let's see here. Last time, let's see.
Dr. Egglin?
Yep.
So if we're reading, are you just wanting us to read the pages that you assign, or do you recommend reading the entire chapter?
I will, once we get going here, I'm going to give you some specific pages where the stuff is so that you can find it easier.
Okay.
For chapter 1, it's pretty self-explanatory because there's not much detail in there.
Yeah.
But like for chapter 6 and chapter 27, I don't know if I got that on Moodle this time or not. Let's see here. These are the pages we're going to do. Chapter, up here. This stuff. And then this is an example of independent learning. So I usually put those in a file that I put on there to differentiate it between the stuff that is the page numbers of the paper.
Okay.
Not that you have to open that up and print it out or anything, but that way you can kind of distinguish between the two.
Okay. So don't worry about anything that's not on those pages.
Right.
Okay.
Right. I'll make sure you know what you're responsible for having to cover, and you don't have to because we just can't do the whole entire chapter. That'd be ridiculous. The other thing about the page numbers is that, and this has driven some people crazy in the past, in order to be efficient and make a decent story out of this stuff, considering we're not going to use 3 semesters to do this book, when we talk about viruses now, today and Monday, we're going to talk about parts of chapter 6 and parts of chapter 27. If I would have written this book, I would have done like an introduction to viruses and the biology of viruses all in one chapter, and then had a section on the diseases caused by viruses. And I'm going to break it up a little bit differently than what these guys did. So we're going to, and that's why there's stuff from chapter 6 and chapter 27 on Moodle for the first week. We're not going to do all of chapter 6 or all of chapter 27. And the text associated with the figures. You know, go to the figure and then it says, see figure 27.27. Read that stuff, which is right next to it, right? Other questions? Those are all good questions. I want you to know what you're supposed to know and do. So, okay. So last time, this is going to drive me nuts when I can't see both at the same time. We talked about scope of microbiology, viruses as being non-living. We talked about historical developments in micro. First one being the first time people were able to see microorganisms, which was in the 1600s. What thing did you ever learn about having that happened in the 1600s? What was another, just to give you kind of a reference in time, what grade school level event do you guys learn about? I don't mean something that happened in grade school, but something you learned about in grade school.
Black Plague?
Well, that was earlier, but that's a good guess.
Finding of North America?
The finding of it?
Yeah, in 1692, isn't it?
1492.
Wow.
So it depends on, no, you got something, no, you're on the right track. You just got 2 things mixed up. What was the other one? Columbus, I mean, okay, so Columbus isn't the best guy in South Dakota. But Columbus came here 1492, and then in the year 1620, the Pilgrims came over, right? Mm-hmm. So this was shortly after the Pilgrims came here, to give you an idea of time frame. So this was still colonial times, or even like earlier than early colonial times. What was going on in the 1860s in the United States? Civil War. This was Civil War time. So during the Civil War, people were starting to get an idea about sterile technique because the story is that they realized that when they sutured up people, when surgeons or whatever doctors were like at the time, sutured up people using cotton in the South, they had problems with infectious disease of the wounds. But if they used horsehair when they didn't have cotton available, they found that they had to boil the horsehair for a long time to make it soft enough to use it for suturing. And those wounds didn't seem to get as infected And why do you suppose that was?
Sterilization.
Because they boiled it and they sterilized it and they cut down on the microbes. The cotton they just took out of the field, it was filthy with spores and all kinds of stuff. Even if it looked white, there was microscopic organisms on there. So they're starting, that's what the kinds of anecdotal things that kind of went into people believing that there were microorganisms that you can see. Germ theory of disease, that's where we left off last time. So the guy associated with that is Robert Koch, German microbiologist. And he not only was the first one to define the causative agents of an infectious disease, more importantly, he came up with a systematic way of demonstrating and determining which organisms cause particular infectious diseases. And that systematic way is what's called Koch's postulates. It's a systematic way of showing that a particular organism causes infectious disease. So there's a lot of limitations on this. But let's just talk a little bit about what the steps were. So this is from a figure in your book, in chapter 1. And these are the 4 postulates, 4 things that have to be true in order to show that a particular microorganism causes disease. So he figured this out with several different diseases, including tuberculosis, including anthrax, including a few others that aren't coming to mind right now. But it was more than just showing what bacteria cause tuberculosis. It was coming up with a way to show that any bacteria causes a particular infectious disease. So these are the ideas here. This shows the model they use. In this case, they're talking about tuberculosis with a patient, a human patient. In order to do things with a human, though, of course, you have to have an animal model, and that's what's shown here with the guinea pigs. So the first postulate is that the microorganism must be present in every case of the disease. So if every person who has tuberculosis has this particular microorganism in them, that's the first step to establishing it. The second thing is that the suspected organism has to be able to be grown in pure culture. So that means it has to be grown in the lab all by itself with no other organisms present in the culture. So you have to be able to get it all by itself. That's the second one. So at the time, people were just working out the techniques for growing microorganisms. You just couldn't go and buy agar and mix it up in the autoclave. He actually had to figure out how to grow them. And he did that using blood serum, coagulated blood serum. So he grew the microbes on there, and then the third postulate is that the same disease must result when the isolated organisms are put into a healthy host. So in this case, he couldn't use a healthy human being, fortunately, although people did back then. We'll talk about that another time. But he didn't. He, unfortunately, had an animal model. And he took the bacteria he got from the person and put it into an animal model and showed that the same disease resulted. The guinea pig got the same symptoms as the human had with the cough and the respiratory problems and stuff. But to finally just demonstrate that the organism you put in first is a causative agent, you have to then take a culture from the animal that has the disease, in this case it actually died, and isolate the organisms from the host, the dead host in this case, and show that you got the same ones back. So Koch isolated M. tuberculosis in pure culture from the dead guinea pig, and we can compare it to this one. So that was important for a few reasons. One is that people could now show what particular organism caused disease, and that allowed people to control disease. It also was really important because it allowed people to, or it forced people, necessity is the mother of invention, right? It forced people to come up with ways to do pure cultures and to grow the organisms and develop animal models, and basically gave a framework for how they're going to even consider doing this. So when would you, well, do you have any questions about how that works? We're going to do a lab activity towards the end of the semester on this. Do you have questions in general about these steps? Christian?
So like, how do you overlay point 3 with the fact that human immunity is so variable, and that like certain organisms cause different outcomes?
That's a very important question. So depending on the kinds of pathogens you're working with, that could be a huge problem. In the case of the diseases that they were studying at the time, and he got lucky that he was choosing diseases that were really big problems. And of course, those are the ones they focused on first. They were typically diseases caused by pathogens that were really virulent, and you had a high likelihood of getting it if you're infected. But you're right, if you chose an organism that wasn't as so virulence, here's a vocabulary word you have to kind of know before you can even go on, even though it's not part of this chapter. Virulence is the ability of a microorganism to cause disease. And it's a quantitative thing, it's not just qualitative. You can quantitate the virulence, and we'll talk about that later, and measure how virulent a particular thing is. Virulence factors are the parts and properties, products, properties, that allow an organism to cause disease. So virulence is the ability of an organism to cause disease. Virulence factors are the things that they use to cause disease. And we're going to talk a lot about a whole bunch of different virulence factors later. But that was a good, Luke, you were going to ask something too.
Yeah, I guess how they determined they had a pure culture?
They did that pretty much the same way we do now, by streaking it out onto agar plates and observing that, and you have to make some assumptions to know you have a pure culture. But you streak them out onto a plate and observe to see that they all the colonies that result look the same as each other. And there's a lot of properties that go into sameness: the size of the colony, the shape of the colony, the roughness, the elevation, the smoothness, the shininess, what did I miss? The color. So it's just based on observation, and it would be something that you could get wrong, because some organisms look the same. So those are good questions. So what other things could go wrong with this? So at the time, he chose diseases that were super important, caused by organisms that were virulent. Under what circumstances would this not work? Because there are times today where people can't use Koch's postulates. Tony?
If someone had the bacteria, or the pathogen, but didn't have the disease, it would not contradict number one.
That's right. If somebody had the pathogen but didn't have a disease, that contradict number one. And that happens sometimes. In the case of carriers, for example, we'll talk later about typhoid Mary Mallon, who was a carrier of disease. She was infected, but she didn't have the disease, and that caused a lot of problems. So if you don't have the disease but you're a carrier, how about having an animal model? Not every organism that causes disease in humans has a decent animal model, and so sometimes you just can't ethically do that. You can you can pay grad students money to get infected, but you can't assume that's going to work. What would be another case where that doesn't work? If it was, yeah?
Well, if
Ben?
the organism is previously infected from a different source.
You mean like in its own historical past?
Yeah.
Like if it has immunity? So it could be that the host is immune, and that would screw things up. That's why statistics would be important. You'd have to know how big of a sample size you had and everything. That's true. It could have immunity. What I was thinking of is that it may not be possible to isolate the organism in pure culture. So some pathogens can't be grown in pure culture all by themselves. Like the organism that causes syphilis, you can't grow that on a Petri plate. We'll talk more about that later. Other things that can't be grown in pure culture like that would be viruses. They have to be grown inside of host cells. Some diseases, like periodontal disease, are caused by multiple pathogens all working together. That's called a polymicrobial disease. And if you have to have a few different organisms together combining all their virulence factors in order to cause disease, you wouldn't be able to figure that out by having a pure culture. You'd have to do some kind of a different model. So there's reasons why this wouldn't necessarily work, but it was super big news and very impactful when they figured this out in the 1800s. Last one. Let's see. Did I tell you already? Yeah, okay, I was going to do 3, but I'm going to do 4 because it's perfect timing. The fourth, super important, according to me, and most other microbiologists, bacterial physiologists, was recognition that microbes are involved in cycling of elements. This is an ecology topic, not that the others aren't in the broadest definition. Recognition that microbes are involved in cycling of elements. So you guys obviously noticed by now that I like to write out the notes and not give out pre-copied printed handouts of the notes, because I think, and I know actually, because people who study this kind of stuff have shown that when you actually write stuff out by hand, it helps with learning and it puts it through your brain. And you might, some of you guys probably didn't write this in exactly the same words I did, because you've already processed it in your head and integrated it and then put it back out in different words, which is good learning for you. And so you've already gone through this once. And in the past, I've had really, really good students tell me they like it that way too, because then they have to, the successful students learn that way too. Okay, recognition that microbes are involved in cycling of elements. There's two people who are really known for that, but the one we're going to talk about today is related to the soil you're collecting, and that is Sergei Winogradsky. And he lived, this guy actually is the most contemporary of these. He lived into the 20th century. He died in 1953, which seems to me like a really late time that people just figured it out. He was doing his work on this in the 1880s also, though. So he's the same. Pasteur, Koch, and Winogradsky were all pretty much at the same time. Pasteur and Koch actually knew of each other, and they actually didn't like each other very much. They had, save that for another day. So what do I mean that they were involved in cycling of the elements? It has to do with biogeochemical cycles, like the nitrogen cycle and the iron cycle. And the big thing that he figured out, the reason they're involved, is that bacteria can use inorganic compounds as their source of electrons. We talked about this a little bit the other day. Bless you. Bacteria can use inorganic compounds as their source of electrons. So the other day, when I brought this up, one of you guys over here said that we usually use carbohydrates as our source of electrons, like sugar, or starch that we break down into sugar. A lot of bacteria can do that, and oftentimes when you learn about bacterial physiology, you do glycolysis in the Krebs cycle and talk all about sugar again. But lots of bacteria don't even like sugar. They don't even take it up, and they don't really have a use for sugar. If they need to have glucose 6-phosphate for making their nucleotides or anything, then they go backwards to make that. But some bacteria don't even take sugar up from the environment. So they use inorganic compounds. So what would be an example? For example, you guys probably learned in chemistry that there's lots of different redox values. Like iron, for example, anything that has electrons to give up could theoretically be a source of electrons for bacteria, a source of energy. So really common ones are H2S, which has electrons to give up, iron, like I said, ammonium, 4+, pretty much any inorganic electron-rich molecule is a potential electron donor for bacteria, for some kind of bacteria, not for the ones necessarily that cause disease, but in the soil they are. And they're responsible for moving inorganic materials through the biogeochemical cycles. And as they do that, they get something out of it. We're going to talk more about that in detail. So he discovered that they use inorganic compounds as a source of electrons. He discovered things like sulfur metabolism. He discovered nitrogen fixation. And we're going to talk in detail about all those. As far as Winogradsky goes, it's important for Tuesday because you guys are going to use the soil you bring in to make a column like this, a Winogradsky column, which is a model ecosystem for bacteria that, in this case, the way we're going to make them, use sulfur as part of their energy generation metabolism. So we're going to take the soil that you bring in, mix it with a source of carbon, and some of the organisms in there will use the carbon as their source of electrons. The carbon we're going to put in is going to be lately we've been using torn up paper towels, right, Libby, which is cellulose, torn and lignin. And we're going to put that in there. We're going to put in some source of sulfate. And what's the other thing we put in there? It's escaping my mind. There's 3 things we put in there, but sulfate. And the sulfate is going to serve as a terminal electron acceptor. This is SO4. That is going to be a terminal electron acceptor for bacteria that do anaerobic respiration. So I think that it's helpful to learn about this stuff if you compare it to metabolism you've learned about before. So in our bodies, what do we use as our terminal electron acceptor in the mitochondria? Oxygen. So these guys are using sulfate instead of oxygen. And when they do that, when this gets reduced eventually, there's a few species along the way, but eventually they get H2S, sulfide, hydrogen sulfide, which serves as an electron source for some of the bacteria that are in here. So those bacteria then can take electrons from sulfide and make it back into sulfate, which some other organisms then use as a terminal electron acceptor. So you have a little cycle going on here. Some of the bacteria that are in here and participating in that are phototrophs. So this is leaking all over the place, so I'm not going to pass it around. In fact, I'm going to get it off of here before I get in trouble. I'm going to set it over here. So when you guys go, you can come up here and look. I had a Winogrodsky column in my office until about July, or maybe August, that was 10 years old, and in an unprecedented move, I cleaned my office and tipped it over and broke it all over everything. And so I had to bring my giant column today. You can't really hand it around, but if you look at this, there's colors of red. Maybe if I put it on the document. There's purple and red and rust-colored. These are my favorite right here. See that? Ooh, that's beautiful. Those are purple sulfur bacteria that use sulfide as their source of electrons. They use sunlight as their energy source, and they make sulfur granules, sulfate. No, actually, they make elemental sulfur, which they keep inside of their cells until it's dark and they don't have enough energy, and then they can use those. The green ones over here, see this yellowish stuff? Those are green sulfur bacteria, and when they use sulfide as their electron source, they generate sulfur granules and they spit them out. So they put sulfur into the environment, and then other organisms have access to it. Down here where it's super black, there's some pockets in here that are really, really black. That's places where sulfide has interacted with iron and made iron sulfide. And so there's iron and the reason there's iron in here is because there's iron in our soil. That's one of the most abundant elements, right? So depending on what soil you bring in, you get all kinds of different things happening in there. We help make sure the sulfur cycle happens so we can see this stuff. Oh, look at this white stuff. We'll talk about that later when we get into the sulfur cycle. So these are beautiful. And some of our students will take these and save them for years. They'll take them off to grad school. Yeah, Libby?
Why do you have so much water in the top of your column?
Well, I did that as an experiment because I wanted to see in some... This interface right here, this is the interface between water, it slips upside down to you, water here and the soil here. I left it that way because I wanted to see if there's any enrichment for organisms up in this region that might be anaerobes. And I don't see any that look like anaerobes, but look at this stuff. This has the characteristic look of this bright bluish green. Oh, it's kind of hard to see. You might want to come up and look at this. I know you're going to want to see this. Bright bluish green is what people used to call blue-green algae. This is actually probably cyanobacteria, or maybe even eukaryotic algae. And one of the reasons I know that is because they look filamentous and the color, but also look at what's happening. I don't know if you see right here. That's a gas bubble that formed between the glass and this big sheet of algae. That's probably oxygen that these guys are making. So up here, it's aerobic. They're doing oxygenic photosynthesis, just like plants making oxygen. Down here, it's really anaerobic, and in fact, they're generating sulfide, which is a reductant, making it even more anaerobic. So there's a gradient through here between totally aerobic up here and totally anaerobic down here. And different organisms grow in different places based on the creation of the environment that they need. And in a perfect Winogrodsky column that's usually smaller, you can see really sharp horizontal lines. This one, because it's all big and I don't know what all, some of them are kind of vertical and patchy. Still very cool. My favorite thing.
I just can't, but the columns that you all will make will have, 2 centimeters of water?
Yep.
So you're not enriching for anaerobes, right?
You're not enriching for aerobes at the top, but you'll still have you'll still have algae and cyanobacteria at the top. And then as these develop, we'll take samples out and look at them under the microscopes, and you're going to love it. You'll love it. You probably want to take them home with you. Okay, I'm going to put that there just so I don't have to worry about that one. That one, the kids who put the mud in, so that column is from one year when we had all the students, I got that at like the dollar store or something, really cheap. And I had all the students put their leftover soil in there. It's the kids who did it are just started med school. So,
No.
is that right? Maybe I'm mixed up. It's 4 years old at least, yeah. So it's not those students, it's 4 years old, probably 4. Years olds develop colors in just a couple weeks. You're going to make small tubes. One year when we did this, the dean's office called me up and said some girl's dad just brought a 5-gallon bucket of mud, and they said it was for biology class. And so you don't need a 5-gallon bucket of mud. Somebody dropped it off in the dean's office. Used. Nice dad, though. Okay, questions about the... Anything? So I want to take a break to do an example. Who's got the clipboard? Is that going around?
Right here.
All right, keep it going. An example of show and tell. Remember, your presentations can be about something you read or heard on the news or something you looked up that you're curious about, or personal experience. If it's personal experience, use good judgment about what you share, right? So I will make an example of myself and my unfortunate incidents in the lab. When I was in grad school, I was working with some bacteria that I had in a syringe, concentrated bacteria. I took a whole bunch of bacteria and centrifuged them down and concentrated them to about 10 to the 10 or 10 to the 12 cells per mil, which is 10 billion cells per mil, something on that order. And I had it in a syringe with a needle on it, and I was trying to stick it into a rubber stopper to set it down, but I accidentally missed and stuck it right into my finger. And it hurt, but it wasn't that big of a deal. It was just a poke. And I pulled it out and went on with my work. And then by the next morning, my finger was so swollen I couldn't bend it. It was red and very painful. And the next day, I couldn't sleep because it was throbbing and it was so painful. So I went to student health, and the doctor diagnosed me with a condition known as infectious flexor tenosynovitis, an infectious disease that I never heard of before. This is take-home point number 1. And that is a disease that results from the penetration of bacteria into the synovial sheath around your tendon. So I want to show you guys, I've never even taken anatomy. I'm a bacterial physiologist, so I don't know much about anatomy, I will admit. So I brought this figure of your synovial sheath. This is a bone. So in this case, this is the bone of my finger right here. This is the tendon. It runs along your bone. And around your tendon, you have this sheath called the synovial sheath. And inside the sheath is synovial fluid that acts as like a lubricating agent for your tendon. And if you get bacteria inside there, you can get a serious infection. And the typical treatment is antibiotics and maybe even surgical drainage. He told me that many people end up hospitalized because of it, and some people even have to have amputations because of it, because it's hard to get antibiotics into that sheath. And I think he's partly trying to scare me into making sure I took all the antibiotics, which is a whole other big thing we'll talk about another day. So I had to be on oral antibiotics. I had to actually get injections of antibiotics, and when I was in grad school, I was about 40 pounds lighter. And so they gave it to me in the butt, and they split it up into both sides because I was so scrawny. It was an insulting day because I had 2 shots, my hand was swollen, I couldn't work, and not only that, I had to keep it elevated like this. And I had to go to work with my hand elevated like that. Anyway, that's take-home point number 1, the existence of that. Number 2 is, I will admit, selfishly I included this take-home point because I want you guys to know this early in the semester. That is the concept of normal microbiota. So in the case of the infection I got, I first thought it might be caused by the bacteria I injected into myself, but I have reasons to think that's not true now. The doctor thought it was probably because I injected a needle through my finger and carried bacteria from my skin into the synovial sheath. So the normal microbiota on my finger actually caused a disease when they got into the wrong body site. Normal microbiota are the bacteria that are normally expected to be found in any site of your body, any particular site. The ones on your skin are not necessarily normal anyplace else. The ones in your large intestine certainly are not. So the normal microbiota in one body site can cause infection when they're introduced into another body site, which is what probably happened with my infectious flexor tenosynovitis. Questions? So these are the take-home points. These are things that I can potentially use to write exam questions. They'll be short exam questions. It's not like it's going to be an essay on somebody's talk, but they're concepts and vocabulary. Notice that I tried to use something that would actually teach you things and not just something like, if you poke yourself with a needle, you can get hurt. You know, I tried to like incorporate some knowledge to make the take-home points a little bit more interesting and something I can actually use. Questions about this content, or questions about what you guys need to do? We can't have them be very long, otherwise with 37 of you guys, all of a sudden we're going to like not have time to do other stuff. Okay, we are now switching to the section on viruses. So we're going to talk about viruses in two separate parts of the semester. We're going to talk about them now in the context of biology of biology of viruses here to introduce what viruses are, and then we're going to come back to them at the end and talk about viral diseases separately when we're talking about pathogenesis and diseases. And I like to separate them from the bacterial diseases because people often get viruses and bacteria mixed up in their heads. You won't anymore after this. And also the diseases caused by viruses and bacteria, so we'll separate it all out. So this is just to do a little reminding about what we're learning. This is on here. Basic biology of viruses. That's now. How they impact human health. That's later. Okay. So we're going to talk about topics from chapter 6 and chapter 27 here. Biology of viruses. So what do you guys know about viruses? We talked about this a little bit on the first day. You know that they cause disease. Some of them cause disease. Not all of them cause disease. Tony, what'd you say? Oh, okay.
They carry genetic information.
They carry genetic information. So they have a genome of some kind. That's one of the things we're going to talk about.
They need a host to live.
They have to have a host to reproduce, for sure. Interesting thing about that is that every potential host has its own set of viruses. So there are viruses that infect humans and all kinds of animals. There are viruses that infect bacteria. They're called bacteriophage. Viruses that infect fungi and plants. Everything's got its own viruses. What else? There's debate about whether they're alive or not. I'd say no. We talked about that last time. They're small in general. So some vocabulary, first of all, in terms of how they're set up. They're non-cellular. So we don't have a viral cell, an individual virus particle. So if you talk about a virus, you might be talking about a species of viruses. But if you're talking about an individual unit, that's called a virion. That's a complete viral particle. It's a viral unit. They call them viral particles. A lot of nomenclature comes from before people really had a good understanding and vocabulary. So they have, in order to be a complete virion, they have their genetic material. They've got a coat, a protein coat. At least. Sometimes they have additional layers that we're going to talk about now. They're typically small. I think the book probably says that they are between 10 and 400 nanometers in size. But recently, I mean just in the last few years, people have discovered some really, really huge viruses. In fact, at first they didn't really know they were viruses. People have seen some now that are more like 700 nanometers in size, which means you'd be able to see them on a regular light microscope if you had a way to stain them. They're huge. And people are still learning more about those. So do you guys have a grip on how big a nanometer is? Last semester I asked this and it was let's just say, how many nanometers are in a micrometer? 1,000. 1,000 nanometers are in a micrometer. So 700 nanometers is the same as 0.7 micrometers. Right? So those are ranges of viruses, and people are still exploring and understanding viruses. The stuff we're going to talk about is just what we know about viruses, not everything there is to know about viruses. So they're acellular organization. other important factors to go into a little bit more detail is their genomes. There's a lot of variability in what their genomes can be. The genomes can have a huge amount of variability. The ones we're going to talk about first have double-stranded DNA genomes, and we're talking about those first because they're conceptually similar to what we have. Some of them have single-stranded RNA, either positive or negative stranded. So with RNA, it's typically not double-stranded. Usually they have the top strand or the bottom strand. Viruses come in either form of what they can have. And the other thing that one of the things you came up with is that they have an obligate requirement for a host cell. In order to replicate. Okay, so I want to talk about some viral structure, parts of viruses. Again, because there's so much variability, here we go. There's a kid eating a hot dog on the first slide. We're not going to talk about that. That's going to be in the disease part. Let's talk about the structure, basic structure of viruses. Some of this is vocabulary and some of this is conceptual because they're different from the cells you've learned about. So the first thing they have is what's called a nucleocapsid. That's the most basic of virion structures. The most basic part of a virus that you can have is a nucleocapsid. And there's a lot of variability on what those could be. There's a picture in your book that shows a bunch of different variety of virus structures. Some of these are what you would call a simple nucleocapsid, and some of them have some other things around them. But in the most basic form, they have nucleic acid, whatever theirs happens to be, and the capsid. The capsid is the protein that protects the nucleic acid. I'm going to abbreviate nucleic acid NA right here. That's nucleic acid. So they come in a few different shapes and forms. This is an example of an icosahedral nucleocapsid. I brought one in here that's a model. And there's a few things to look at with this model. An icosahedral nucleocapsid is one that is basically shaped like this. It's got 12 corners, 12 vertices, and 20 equilateral triangles making it up. And you can try to count it. It's not very easy to count them. But that's typically the way they come together. Some viruses don't have anything more than this and the nucleic acids that go inside of them. Some of them have other parts that we're going to talk about in a little bit. I'm going to pass this around and we're going to talk later about some of the properties of the pieces on there. So when you're looking at it, think about how they fit together. And in this model, there's two different colors of proteins. These individual things represent a single polypeptide. Brandon?
Slightly off topic, but
Sorry.
Apart from a host, can viruses exist like in a dormant state?
Yeah, and different ones last for different amounts of time. Some of them, I mean, imagine if they're transferred from host to host, they have to leave one host and get to another, and they do that outside of the cells. Depending on the structure of the viruses, some of them can last for a long time, and some of them are pretty fragile. But they definitely do. So those, okay, so that's an example of an icosahedral. I'm going to write that word down. I'm having a hard time saying it. Icosahedral. Another shape, helical. This is a little different from the icosahedral because in that case you build that shell and the DNA gets, or the RNA, whatever it is, gets packed inside of there. In the case of the helical viruses, it's a hollow cylinder. And there's a picture of it there that I've got up on the screen. And the hollow cylinder is made up of protein subunits. In that case, the protein subunits are the triangles, the yellow and the red triangles. In this case, the protein subunits are shown like this. They're these individual, they're called protomers. They're all the same as each other, and they come together and make this hollow cylinder, and the genome is inside of the hollow cylinder. So to give you an idea, in this case, it's about 20 nanometers wide. They're really small and skinny. They might be long, but they're very small, or very narrow. So in some cases, in terms of structure, the viruses, either one of these, can have an envelope around them. So the envelope is a membrane. Remember the last time we talked about whether living things have a membrane and whether there's an electron potential? In some cases, they have an outer membrane that's made out of the host cell's plasma membrane. So imagine, we're going to talk about how this happens later, but what happens is that the virus, that virion or nucleocapsid, is leaving the host cell and it buds off, and as it comes out, it leaves and takes with it part of the host cell's plasma membrane, and it gives you something that looks like cartoony, like this. That's what is represented here. Here's the icosahedral structure, and then this is the envelope that's made out of the membrane from the host. So there are some that fit into a fourth category that aren't all, they're not this simple. They're just called the complex. That's what they're called, the complex viruses. And they're neither icosahedral nor are they helical, and they're not made out of a single type of peptide. In the case of these, each of these triangles, there's a relatively small number of triangles, but they have a similar enough straight shape that they can all come together in that form. Same thing is true of these guys. But in some cases, you can have viruses that are considered complex, like this one. This is a bacteriophage. It's got a kind of protein here, it's got an icosahedral thing up here, it's got these tail fibers and a different kind of plate. The whole thing is made up of so many different parts that it doesn't fit into the other categories. That's what you would call a complex virus. Okay, Hannah.
Is the envelope how some viruses like avoid the immune system in other hosts?
That's a good question. Well, it's sort of the, if all they had was the envelope and there's nothing in there besides the plasma membrane, they wouldn't be detectable probably, because we don't have a huge amount of diversity of our phospholipids, you know what I mean? We have a bunch of other carbohydrates and stuff sticking out there that we would be able to detect. But actually what the situation is that outside the envelope, and we're going to talk about this right now, they have other structures like these, and where's the other one I was just looking at? This one, this is influenza virus. These green things sticking out here are part of what our immune system recognizes. So we don't recognize the plasma membrane, the phospholipids, we recognize the proteins that get stuck out there. So if they didn't have those proteins, they would have less virulence because they wouldn't be able to, but we'll talk about the role of those in a second. Okay, other questions about basic structure? All right, we're not going to get through, we're going to, 5 perfectly good minutes here. Okay, specific parts. Viral parts. One are the protomers that we just were referring to. Those are the protein subunits that make up the capsid. So where'd that thing go? It's the red and the yellow things on that structure. Protein subunits in the capsid. And in the icosahedral and the helicoviruses, there's either a single or just a few different proteins that do that. So that one shows 2. It could have been just 1. And the reason for that is conservation of genetic material. If they can get away. So the protomer, this is just a protective structure. These proteins are going together to make a protective structure. And one of the things about the viruses that we'll talk about is how small their genomes are and how efficient they have to be with their genomes. So they could have a whole bunch of different examples of protomers that put it all together, but they get away with as little as they can so they can save their genetic material. One of the really cool things about them is that they self-assemble to make the capsid. Meaning that they're not processed. It's not as if they go through the Golgi and something happens, or that there's some kind of machinery that puts them together. All of the information needed for them to polymerize like that is part of the structure. So this is an example of structure and function. Remember the core concepts that we talked about, structure and function? the function of this is dependent on the shape of those things because they all come together spontaneously and assemble by themselves, not because they're put together. And then that also goes back to the efficiency of the genome. If they had to carry all the machine with them to put this thing together, they would never be able to stay small. Tony? Pardon me? The codemers, they are encoded by the viral gene. Yep. So the protomers are the proteins that are encoded by the viral genome. So the viral genes get expressed, they get yep. And then the proteins that are made, it's sort of like crystallizing. It's sort of like they crystallize on themselves. It's not crystal formation because it's different, but they have affinity for themselves. And so they form this structure without having to be built by anything. Does that make sense? No outside from any other kinds of molecules. Okay, one more thing, and then we're going to have to carry on later. Other thing that they all have to have, nucleic acids. That's their genome. The size, they try to stay small. Generally, we say they're between 3 and 380 kilobases. So 3 kilobases, that's in a eukaryotic cell, that's, well, let's say in a bacterium, that's barely enough for 3 genes. That's really small. So their DNA can be, or their genome can be single-stranded or double-stranded DNA. It can be single-stranded or double-stranded RNA. And of course, because it could be single-stranded, it could either be plus or minus stranded, meaning either the coding strand or the complement of the coding strand. So the positive strand, when you talk about positive single-stranded RNA, this would be a good thing for you guys to think about this weekend before we come back on Wednesday, because this is where people get confused. The most confusing thing about understanding a little bit about viruses is about the RNA viruses, because they have RNA genomes, and that's different from what we're used to thinking about. The positive single-stranded RNA is the coding strand. It's essentially the mRNA, right? The negative single-stranded RNA is the complement of the coding strand. So the mRNA, so ordinarily when we think about human cells, we have the DNA, it's transcribed into mRNA, and that is the information needed by the ribosomes to make the proteins, right? So in the case of the negative single-stranded RNA, the product of the DNA would not be helpful for making would not be helpful for their genome. They have to have the opposite of that. They have to have the complement of the mRNA. So that is, let me tell you guys, I'll do you guys a favor and show you. This is in chapter 27. If you're going to do any microbiology this weekend, you'll do microbiology for sure, whether it's related to this class or not. I would look at these two figures. Figure 2723. This shows a positive RNA genome. And it tells you about how that gets replicated using the negative RNA. And this is the negative RNA genome down here. If that's your genome, in order to make protein, you have to make the positive strand. That's what the ribosomes recognize, right? The viruses are going to use our ribosomes. They have to have mRNA, so this gets used as a template to make mRNA, or I should say positive RNA, positive in both cases. That is used as a template for making the protein, and it's also used as a template for making more negative RNA that's going to be their genome. So in order for the virus to reproduce, it has to make more of whatever its genome is. And if its genome is negative stranded RNA, it has to make positive in order to use that as a template to make more. Of course, we're going to talk about this in class, but if you look this stuff over in chapter 27, chapter 26, or chapter 6 is its parts and things to learn. This is more concepts and how things work. So I would spend more time on chapter 27. Questions?
Lab attire.
Oh, lab attire. Closed-toe shoes, long pants. It's a new day in town. We're trying to be safe. It used to be the Wild West.